Hello.
Hi.
Hi, how are you?
Pretty good. I just wanted to tell you I probably won't make the next meeting.
Okay.
Going on a holiday.
Oh, yeah? That's good.
Yeah. So,
Sutrik and Aniketh, hello.
Yeah. Hey.
Hi, how are you?
Good morning. Hi, hello everyone. Great.
There's Morgan. Hello. So I don't know if anyone, I thought maybe one of the aspiring GSOC students wanted to present today. I didn't know if that.
So
Yeah.
I'm an aspiring GSOC participant.
Okay, yeah. But I mean, I had someone in mind, and I don't know if they want to present this week. I don't know if they're here. I can't remember who it was, but if they want to present, that's great. Well, I'll do some stuff first, and then we'll circle back around to that. So, but I'm going to present something on Synticial and C. elegans today, and then here's Mehul. And then I'm going to talk about some other things as well. So, actually, let me ask Mehul. Mehul, did you want to present something?
Hi Bradley. Yeah, so I don't have anything to present, but I was talking to Joy. He wanted to present this transaction graph. But he told me that today he's not well. Like from yesterday, he's not well, and he won't be able to join. So next week he has a presentation ready, and yeah, so that was his plan.
Well, yeah, all right, that's good. Thanks for that. Yeah, I'm looking forward to it. Transaction graphs, you say?
I think he called about this. I don't remember exactly. It was temporal graph, not transaction graph. I'm sorry. Yeah.
Oh yeah.
So he has good literature review over it. He has one published paper in that, I think.
All right. Yeah. Okay. I look forward to that. So that may be next week. So yeah. I don't know. Yeah, we'll see. Yeah. So that's good. Yeah, we'll see about that. Looks like we have a new person, Pierre Gallipo. So hello. I don't know if you wanted to introduce yourself in the chat. We have a chat function at the bottom where you can type in an introduction if you'd like. And it looks like Jesse's here. Hello, Jesse. Jesse hasn't been here. I have a couple things today. The first thing is we talked last week about graphs and about layouts. And there are all sorts of layouts you can use for graphs. You can use force layouts, force directed layouts, circular layouts, different types of sort of topological layouts where you have like space that's taken into account. This is from a package called Gephi. I don't know how many of you are familiar with it. This is something that is made kind of for graphs. This is made for graphs for plotting them. You put them in the form of your nodes and your edges, and then you plot them out, in some sort of layout so you can gather statistics and all this. And it's in a graphical user interface. And this is from the documentation for Gephi. So this is where they guide you through a tutorial on layouts. So this is an introductory tutorial about layouts in Gephi. It will guide you to the basic and advanced layout settings in Gephi. You will learn how to use various layouts according to the feature you want to emphasize in the topology and size of the network. So this is where they have built-in topologies for networks. They have a built-in circular layout. They have a geo layout, which means you can plot them in, I think, geographic space or some sort of geometric space. There's some metric space they use. You can also have a geometric transformation, and there are other ones as well. Their force layout, I know I've used those. where it's like they use sort of like they just put the nodes in the optimal locations where they're not overlapping and you don't see a bunch of overlap in the layout. So it's like it might be like, you know, laying it out on an axis or laying it out in clusters or whatever. And those don't mean anything other than that's where it's physically optimal to lay those nodes out. So they have a number of different, so they have Fruchterman Rheingold, Force Atlas 2, they have the circular layout, the radial axis layout, geographic map, and, you know, various other ones. And I think you can write your own, one of the reasons I bring this up is I think you can write your own plugins for this. So Gephi, of course, runs, it's an open source tool and it runs from a GUI, but you can write extensions and plugins and you can do interesting things with that. I've generally used Gephi for a lot of the stuff I've done. It's maybe not optimal. There's also, you know, tools, JavaScript tools. There are also Python tools, I believe. But for visualizing things, Gephi works fairly well. So that's just that tool. Okay, now I want to get into this Synticial and development. So I did I think last week I mentioned that I had collected some papers on, and this was an original question from Dick Gordon about Synticial and C. elegans in development. So Synticial are cells that have multiple nuclei, and they're usually the product of cell fusion. And the question he asked was, do these show up in the lineage tree? And if so, can you put them in a lineage tree? And so the answer that I found, the short answer, I'll go through some slides, but the short answer is I don't think that people have really put that into lineage trees. You could do that, but there are a lot of actually, as you get into the literature, it's both unclear as to how to represent them, and then like what basically visualizing their larger significance and development. There's a lot of things that Synticial cells do and where they are in the anatomy that are really interesting. So I have a chat message here. Okay, yeah. Welcome, Pierre. Yeah. So this is Pierre's introduction. Pierre Gallipo. Good afternoon, morning, everyone. Sorry, I have another talk I promised to attend, so I'll try to follow both. I'm a postdoc specialized in morphogenesis, and I've been in contact with Dick for a few days concerning a project to structurate the morphogenesis domain. Welcome, Pierre. Yeah, this is our group. We've been doing this group for about 10 years, a little over 10 years. And, you know, we talk about developmental biology and biophysics and computational biology. So we have a lot of fingers in a lot of things. And so we'll talk about like machine learning stuff. We'll talk about developmental biology stuff. We'll talk about a lot of other things. So welcome, and we'll see. We'll see what you find interesting, and please let us know if you find something interesting. Okay, so I'm going to start this presentation, and this is Synticial in the C. elegans lineage tree. So Synticial cells are these cells that have multiple nuclei. We'll see some microscopy of what they look like. I was trying to figure out how to draw them out, and I don't know if I would get it right if I drew it out. I mean, I know what the microscopy looks like, but I was thinking of drawing some sort of time lapse or time series pictures, and I couldn't do it. So maybe I'll do that later. But this is just kind of an overview of the few papers in the literature, so that we can see what they have just in C. elegans. So this, of course, is our C. elegans lineage trees. You see, we're at an advantage when we work with C. elegans because we have, cells that divide, and then we have a nomenclature for each sublineage, and those cells are invariant, meaning that they don't change their identity in development. So when you have, say, for example, a zygote, it divides into two cells, an AB cell and a P1 cell. that P1 cell has a number of deterministic fates, and the AB cell has a number of deterministic fates. And if you remove some of these cells in development, if you ablate them out of the embryo, then the worm will be dysfunctional in some way. You have mutant phenotypes that have missing cells, and mutation acts upon the lineage tree as the cells are dividing. It just doesn't, either it divides in a asymmetric way or it doesn't divide, and you end up with these. There's no compensation like you might see in mammals. All the cells have a known role, deterministic, and the organism is what we call eutelic, which means it has a finite number of adult cells that it ends up with. So that's the context for Synticial cells in C. elegans. You have to have a certain number. You always have a certain number of cells in an adult, either if it's a hermaphrodite or a male, and the mutants have a certain number of cells usually. So if you have a mutant that's at a certain allele, that mutation then results in a certain loss of cells. So everything is very regular. So it's a good system to study the Synticial cell process, formation process. So this is the lineage tree again. We're organized from anterior to posterior. So AB, that side is anterior, the P is posterior, and all these other sublineages are different specialized sublineages with, that end up with different types of tissues. So this figure here on the top, you see the blue, which are endodermal cells. So sublineage E results in endodermal cells, D results in mesodermal cells, P4 is the germline, C is a mix of neurons, endodermal, or neurons, mesodermal, and hypodermal cells or epithelial cells. And then MS is a mix of mesodermal cells and neurons, and I guess I guess it's mesodermal and endodermal cells. Mesodermal and neurons. So yeah, the point is that there's this, we know what every cell is. And so down here at the bottom right, or the bottom left, you see that there's this axial organization of the lineage tree, anterior to posterior, and then also right to left. If you notice our nomenclature, the A's are anterior, the P's are posterior, the L's are left, and the R's are right. So you can pretty much know where a cell will end up by its nomenclature. And you also see in the lineage tree that if it's drawn in a certain way, this is from Worm Book, you get these cell migrations that happen across the anatomy. So the cells will move around, their origins are in a certain part, and they'll migrate to certain places. And you get these, this is a special interesting part of the AB sublineage, where you get these repeated sublineages that kind of migrate. And so these, the lineage tree crosses location here. But that's actually kind of important for Synticial cells. So this is Synticial cells in C. elegans. This is just in C. elegans. There are Synticial cells in all sorts of organisms, in Drosophila, in vertebrates, of course, and all sorts of tissues form these Synticial cells. Synticial cells have a common cytoplasm, and so you might have a bunch of different nuclei with a common cytoplasm. They usually have these intracellular bridges. So this is an example on the right of what they call the rachis. This is in C. elegans. And this is your shared cytoplasm. So, we have here, this is from this paper, Goldstein, where they talk about this result. It's basically showing the rachis, where right here, where you have nuclei in a common cytoplasm. And so this isn't just the only type of Synticial cell. Muscle cells are also, or muscle tissues are also Synticial cells, where you have multiple nuclei in a common cytoplasm. So this is, of course, results from cell fusion, of course you might have guessed, and other types of processes that bring these cells together and form these cellular bridges and other types of ways to share cytoplasm. So Synticial cells have a common cytoplasm. They have they're single large cells with multiple nuclei, as you see with the Rachis. And then efficient distribution of cellular components. So the cellular components are distributed within the Synticial structure. Okay, so now this is, of course, I said that cell fusions were important. This is a paper from Developmental Biology, Cell Fusions in Developing Epithelium, or Epithelium of C. elegans. This is where they talk about epithelial fusion. So we had some epithelial cells in our lineage tree, and those epithelial cells will fuse, and they'll form different structures. They'll have different functions. So in this case, they kind of talk about how you see this in the hypodermis, where you see it in vulva, uterus, excretory gland, germ cell formation, and pharyngeal muscles. So all these different types of tissue, usually it's in the germline or in some of the other areas in the worm, and then of course the pharyngeal muscles. The pharyngium is a structure towards the front of the C. elegans, where there's a ring that's formed, and that ring has these fusions that result in these syntitia. Okay, so then there's also something called epithelial adherens, which are cell-cell fusions in embryonic and post-embryonic development. So these are epithelial adherens, where you get these cell-cell fusions, and this is, of course, in the embryo, but also in post-embryonic development. And then we have this, so cells, to do this, they'll migrate around, and they will migrate from embryonic development to post-embryonic development to the adult phenotype, which we know it from, when we see a lineage tree, it's usually the developmental sort of lineage, and then you see the adult cell as the output. But actually, there's there's a significant amount of lineage tree that comes out of post-embryonic development, and you get a lot of cell migration and plasticity before you reach adulthood. So these cells will migrate, they'll fuse in post-embryonic development. So the final positions in the adult phenotype, the boundaries and the sizes of these syntitia are all similar across organisms. So as you might have guessed, in C. elegans, we have this common, we kind of know where a cell will be in development. We kind of know where cells are in adulthood, and there's no difference in the syntitia. They end up in these sort of the same positions as we might expect. There's also this issue of variation in the order of fusion events in lineage trees. So if we look at a lineage tree, cells will migrate, they'll start to fuse, but there isn't like this deterministic fusing order. So one cell doesn't fuse to another cell, and then another cell fuses to another. There's no recipe for that. The cells will fuse, but they'll fuse maybe in their own order, or they'll migrate around and they'll kind of fuse on their own time. There's variation across C. elegans in that regard. What's interesting is that this process is initiated in the anterior portion of the embryo, so towards the left-hand side of the lineage tree, and then proceeds posteriorly or towards the right-hand side of that lineage tree. So that's an interesting point. And it may have to do with the tissues where the syntitia forms. So it could be that you see it in the tissues that result from the AB sublineage first, and then later on, we talked about how the germline is at the far right end of the lineage tree, or the posterior end of the lineage tree. So that may have something to do with it. There also, if we start to think about modeling syntical cells, we have these dynamics we have to think about. And this is a figure from this paper where they talk about this membrane protein that's essential for developmental cell fusion, but they show in this paper, they show the adult here, which is laying an egg. The egg forms an embryo, and you see these cells, which are differentiated developmental cells. So you have this developmental cell lineage. It produces these differentiated cells. So this is a hypodermal cell 6. These are hypodermal cell 7. And then those cells will go on to form these vulval rings. And those cells will either fuse or they're unfused. So some cells are committed to a fusion fate, and some cells are unfused or have that unfused fate. So you can see in this structure as it forms in the vulval rings, you go from the embryo and you have these candidate cells, and then they form these rings. These are toroidal structures, and you can see that they stretch out like this, and sometimes they fuse and sometimes they don't. And as we go through to the L4 stage of um, the L4 larval stage, we usually get this gradual fusion into these vulval rings, and then we end up with this pharynx structure towards the anterior end of the animal. And so you see this pharynx structure. These are these vulval rings in the pharynx structure. They form the ring. They not only go through this differentiation process, but they migrate and fuse to form this ring. That's how it works. And so there are two important things here. There's timing. So we have to model the timing, which is a sequence of migration events, a sequence of fusion events, and then the fusion itself, which is in the cell. And I'm just showing you a cartoon of the cell, but in the cells, you have this rearrangement of adherence junctions and its location in the anatomy, in this case, in the vulval ring, which will end up in the anterior portion of the organism. And you can see that in the embryo, where you get these hypodermal 7 cells that need to move over to near hypodermal 6 and form this ring. This is another paper from Bauer, the initial expansion of the C. elegans Synticial germline. It's coupled to incomplete primordial germ cell cytokinesis. So this is where the Synticial germline, so in the germline we have these Synticial structures that basically have to do, I think, with cytokinesis. So this is where we have this diagram. We have this 2-cell Synticium at the left. We have the cytokinetic ring. We have the cell division event. And then we have where they're kind of fused together. So you have 2-cell Synticium, cytokinesis, cytokinetic ring stabilization. So the ring forms, it stabilizes. You get these Synticial cells as a product of cell division, incomplete cell division, I guess. Cytokinetic ring displacement, and then cytokinetic integration, where you have a 3-cell Synticium. So you get these Synticium that grow in terms of their nuclear material or their nucleus. So you go from having 2 nuclei to 3. You get this incomplete cell division, and you maintain this Synticium throughout. So this is an interesting structure. I'm not really familiar with this sort of. They talk about the germ cell Synticium have been studied a lot. So that's a model system within C. elegans for that. Then this is the same paper, and this is just showing difference. We have the embryo here, we have the L1 worm, and then we have the adult. This shows that there are these different morphologies for each of these stages of life history. And this shows kind of where these are through showing this microscopy data here, where you have these structures. You can see it at A, B, and C. And then the row below, it's highlighted, so it's color with false color. You can see the structures where the cells divide, but they retain a common cytoplasm, but they have two nuclei, and then they have one nucleus here. So you can see like their differences in the number of nuclei, but they all share this common cytoplasm. Okay, this paper is Born and Weiss. This is an interesting paper because what they do in this paper is they take an embryo and they constrain it artificially, mechanically, and they want to see if they can form Synticial cells where in a normal C. elegans embryo you wouldn't see Synticial cells. So this is Born and Weiss, robust spatiotemporal organization of mitotic events in mechanically perturbed C. elegans embryos. This is from Biophysical Journal last year. This is the figure up here where they show like these cell division events and they show the nuclei. So this at the top is sort of the normal process of development, where you get these get these membranes that separate out the cytoplasm between each nucleus. So you can see that this has 4 cells, 4 nuclei. Actually, it says 5 nuclei, and you get basically 1 nucleus per cell, roughly. In the experiments that they do, they constrain these embryos in terms of their size. They introduce physical stresses, and they end up with these Synticial structures where you get multiple nuclei. Basically, the nuclei divide, but you end up with a single cell or just a few cells where these nuclei share cytoplasm. And so that's basically what they're doing here. So they do this volume compression of embryos, and that's their perturbation that they introduce to sort of constrain the embryo. So they do is they suppress cytokinesis, and it forms the Synticium, which is pre-gastrulation. So gastrulation is where you get this these first pattern formation. They get the first pangs of pattern formation, basically. And so, I guess in gastrulation is when a lot of the Synticium start to appear in the germline. So we're trying to induce this before we get any natural Synticium. So the Synticial state is still features mitotic events in multiple rounds of nucleus duplications, as we saw in here. So the nuclei divide. It's just that the cell membranes aren't dividing into individual compartments. And then we have, if we look at the unperturbed embryos, we can look at the lifetimes of Synticial nuclei between successive anaphases, which are these stages of cell division, and these lifetimes increase with every generation. So we see that, there's this pattern that we can observe in the unperturbed embryos that are a little bit different than the ones we get in the perturbed embryos. But we get these nuclei that divide, but not the membranes. So this is where they show some data for the artificial Synticium, which are these squeezed embryos, versus somatic, and then versus germline cells. So I mentioned that in the germline cells, we see these Synticium. Of course, this is pre-gastrulation, but that would be the place where you would see them first. So we have the Synticium here, the somatic cells in the P1, which is the origin. So you can see here in the lineage tree, you have P1, P2, P3, and then you get P4, and then that's where you get like your Z cells, which is this germline. And then all the other somatic cells are out here, A, B, E, M, S, C, and D. So these are all kind of, these are all the sort of the cells that we're interested in outside of the germline. This is the sort of the germline precursors. And so you can see that this is in sort of this tau parameter in minutes. And then for each generation, for each cell division, basically, we're looking at these three categories. And we see that Synticium have this increasing tau in terms of time. And, in P1, P2, P3, P4, it increases. And it also increases in the Synticium. And in somatic cells, it kind of increases, but mostly the variance increases. So yeah, that's not, without getting into the paper too much, that's some data that they show. They have they kind of work out some of these data, and they just show kind of the trends between their artificially induced Synticium and the sort of the precursors of normal Synticium. Okay, so then finally there's this paper, Linehan, lessons on the force-form function connection in cell biology for modeling a Synticial germline. So this is where they talk about this force-form function connection. And so this force-form function connection is basically where you have forces acting, it results in form, and then that results in function. And so there's this connection between those three things. And so that's something that they're looking at here. It's kind of an extension of what people, the same kind of similar technique they were using in the last paper. So their first point here is that Synticium creates exceptionally large cells, dramatic enlargement of cell compartment to support morphogenesis. So when you get a Synticium, it's this very large cell. There's this dramatic enlargement of the cell compartment. It's supporting morphogenesis because you have these structures that kind of can result. And, you can have different, and I don't know, I guess what they're getting at here is that it can serve a function like a muscle or something in the germline. It can also result in, sort of the precursors for morphogenesis. This is why we're looking pre-gastrulation, some of this stuff. Then we have intricate multi-compartment architectures where you have this cortical contact, cytoplasmic flows, and germline extrinsic forces. So you get this sort of, in these Synticium, you get this multi-compartment architecture, even though you have a single cell with multiple nuclei. And then you have this where the germline, which is one example of a syncytium, is because they're modeling syncytial germline. There's this deformation and displacement. And so this is sort of analogous to local and global force production. So the force production from the formation of the syncytium have local and global effects. Okay. So that's all I had for that. Let's take a look at our chat. So we have, let's see. Okay, it's also spelled syncytium, also called mosaic. And then Mehul that was about like the idea of syncytial cells. Mehul says this looks quite useful for the project, and he'll go through the worm book and meeting recording. Thank you for attending, Mehul. And then Dick says, I recently learned that some people regard all tissues as syncytial via gap junctions, despite each cell in a tissue having just one nucleus. Yeah, I think that's an interesting point. Something I noticed when I was reading through the papers. They talked about these sort of connections that form between cells. And you saw that sort of in the germline, where you kind of had this connection. There was like incomplete cell division. And so you had these kind of blobs that had like maybe a ring remnant, and then you cytoplasmic ring, and then you had like the division of the nuclei. So the nuclei were kind of in their own semi-compartment. So, I mean, let me see if I can draw this out. You'll have a cell here. We'll just start on this side. The nucleus, here's the cell. Then you'll get like this ring that forms. You get these nuclei that divide. The ring is here. And then the ring stops. So you get this sort of peanut-shaped syncytium with two nuclei, and then maybe two more as that cell divides. Now, what's interesting is that you could also have a situation where you have multiple cells. Like I said, we can have multiple developmental cells. This. Okay, they kind of migrate towards one another. And then they form these connections. So, of course, gap junctions are these physical connections between cells. And so you end up with these intercellular bridges, which are kind of like, I guess it could be gap junctions. Could be other things too. But you would end up with a syncytium. Technically, it's all sharing the same cytoplasm, but it's much more compartmentalized with multiple nuclei. And then these cells, say, could divide up like, we saw in the last example, where you'd have something like this. This cell would kind of incompletely divide with the ring here. And then you have this shared cytoplasm, right, like that. And you have your complement of nuclei. And of course, we haven't talked about the organelles, which are in here. So you have mitochondria and you have other things. Mitochondria are interesting because, like in muscle cells, those are quite important, right? Because you're trying to produce force, you need to have ATP and all that. So that's kind of what I'm thinking about for how this was represented in the literature. And so, at least explain to me. I don't know. That's what I was trying to struggle with, was how to draw that out. Okay, so it says each gap junction only lets molecules through up to a maximum size. Yeah, not all cytoplasm. So yeah, gap junctions are usually like electrical connections, in the sense that you have like I guess maybe ions or something going through them. So you could have something, you could have something like gap junctions as well. Yeah, also dimolecules. Yeah. Okay. Yeah. So that's good. Any questions? Any other questions about that? So yeah, I hope that was useful. I might try to work out some of that more. Like I said, with respect to the lineage tree, which was kind of the original question, I guess you could like pick something and like look at the lineage tree, because we have the interactive lineage tree in C. elegans as well. So you can go through that and look at like the data that they have on. Actually going into like some of the muscle cells would be kind of interesting, and what those kind of look like. I suspect that the way you would model it on a lineage tree, or maybe like in a differentiation tree, is to kind of model the cell divisions and then model where they kind of come together and just kind of estimate where they come together in development. I don't know if we have the timing down in the embryo, say, or even in the post-embryonic organism. But yeah, it'd be interesting to maybe even create some simulations where you say these cells fuse into a Synticial here. What does that look like? Well, anyways, that's Synticial. Okay, I'll talk about this paper to finish off. This is from the Journal of Physics A. This is not developmental biology so much as it is this idea of Laplacian thermostats. So this is kind of like thinking about collective behavior and some physics. So this is going to be a little bit difficult to get through, I think, but we'll see how we do. So this is a paper, Discrete Laplacian Thermostat for Flocks and Swarms, the fully conserved inertial spin model. So this is by a group of physicists, and they're going to talk about swarms in the sense of bird flocks and other types of swarms that we see in insects and other organisms that exhibit collective behavior. So the abstract reads, experiments on bird flocks and midge swarms reveal that these natural systems are well described by an active theory in which conservation laws play a crucial role. And so we talked about like soft active materials and the role in embryogenesis, but also the role of those kind of theories in describing collective behaviors and emergence. So this is kind of, it's kind of a physics paper, but it'll be relevant to all those areas. By building a symplectic structure that couples the particles' velocities to the generator of their inertial rotations, or spin, the inertial spin model, or ISM, reinstates a second-order temporal dynamics that captures many phenomenological traits of flocks and swarms. So this is a spin model. So it's like spin glass. We talked about spin glasses, and this is kind of similar to that. So they're basically looking at particle velocities and then the spin of these elements. And so what they're trying to do is they're trying to capture the properties of flocks and swarms and their behavior and model them with this inertial spin model. The reversible structure of the ISM predicts that the total spin is a constant of motion, the central conservation law responsible for all the novel dynamical features of the model. So I think in our Saturday meeting, which is it's a separate meeting, but we kind of talk about things that overlap with this meeting. I think it was Morgan and myself, we were talking about emergence and how in emergence, a lot of times you'll take a swarm of birds where they're flocking and they exhibit collective behaviors. They exhibit, the flock has different patterns, and the individual birds, don't engineer that from sort of a master plan. They kind of just interact, and the pattern forms from the interactions, and that's emergence. And one of the properties of emergence is where you can't reverse engineer an emergent flock. If a flock forms a pattern, a coherent pattern, that pattern is a product of their interactions. And you can't reverse engineer that to any one set of birds doing any one set of things at any specific time. Those patterns happen sort of as a consequence of gross interactions. So there are a lot of different ways to get to that pattern. And so this is kind of interesting that there's this reversible structure of this internal spin model. So this is nice because you can have this model. And then on Saturday, we talked about another way that people were doing this to get the sort of reversibility. However, fluctuations and dissipation introduced in the original model to make it relax violate the spin conservation law, so that the ISM aligns with the biophysical phenomenology only within finite size regimes, beyond which the overdamped characteristics of the Vichek model takes over. Here, we introduce a novel version of the ISM, in which the irreversible terms needed to relax the dynamics strictly respect conservation of the spin. We perform a numerical investigation of the fully conservative model, exploring both the fixed network case, which belongs to the equilibrium class of model G, and the active case, characterized by self-propulsion of the agents and an out-of-equilibrium reshuffling of the underlying interaction. Our simulations not only capture the correct spin wave phenomenology of the ordered phase, but they also yield dynamical critical exponents in the near ordering phase that agree very well with the theoretical predictions we make. So this is where they talk about this Laplacian thermostat. So I'm going to get into that in a minute. I don't know, there might be some more items in the chat here. So this is where do birds spin while flying? That might be a good question. I don't know. So this is the introduction here. So approximately 3 decades ago, this paper was from 2024, so probably in the 90s. Vichek et al. Enlisted a sizable number of unwilling statistical physicists into the fast-growing field of biophysics by introducing a simple little model that elegantly captured the key traits of collective animal behavior. This model, partly rooted in the physics of ferromagnets and critical phenomena, partly drawing from inspiration from previous efforts in the field of computer graphics. So they're referring to spin glasses and then also boids, which was, of course, the paper by Reynolds. This is, I think that's what this citation is. Yeah, Reynolds, boids, and then this paper by Vichek. I think this is one of the original spin glass papers, or an early iteration of this work. Okay. Bringing these two things together demonstrated that local alignment could induce collective motion and long-range correlations even in active systems. So we have this long-range correlation, which is where you have correlations between something, say, in the posterior part of the embryo and the anterior part of the embryo, or something in the front of the flock and the back of the flock. Usually, if you have something that isn't sort of this emergent collective behavior, you might have like every bird doing a random walk or every bird in it for themselves. And you can imagine how chaotic that would look from the outside. They'd just be kind of like, it would kind of be like static on a television set, or like this sort of gurgling, percolating mass of particles. And so that's where you have no correlation, or maybe very short range correlations. Long range correlations are where the things that are happening at the back of the flock affect the things that are happening in the front of the flock, or things that are happening anteriorly also happen posteriorly, and there's a correlation. So in equilibrium physics, it really prevents long-range ordering. Under more than one respect, but not all respects, this was the start of active matter physics. Of course, we've talked about the stuff that Desjardins started in the 60s, and then moving forward to soft active materials and some of these other areas, active matter physics, where people are doing things with developmental biology, but also with material science, and even using their models in complexity theory. Vichek's models are truly remarkable for their simplicity and great generality, so much so that they are considered the Ising model of active matter. So the Vichek model is the sort of the Ising spin glass model of active matter, where the Ising spin glass model, of course, is where you have ferromagnets that spin, and you measure the spin, and then there's this, you can build networks that exhibit this long-range correlation. But in active matter, you need to treat it a little bit differently, hence the Vichek. However, as soon as experimental data on actual biological groups started to flow in, it was clear that the quantitative description of certain biological features required models to incorporate some additional elements. So among the many variants and evolutions of the Vichek model aimed at better describing biological reality, one area of particular interest for this paper is this inertial spin model, or ISM. So if you look at starling flocks, you see that the collective behavior of starling flocks, especially during turns, if you've ever watched bird flocks, they're kind of coordinated in their movement. They're all kind of moving in the same direction, and then they make these really complex turns. And that's really where the coordination becomes apparent, because they make these turns and they're kind of these really complex waves almost that they form. So as they follow each other around these turns, they form these waves, but they maintain their coherence. So that's really the thing that's really interesting, I think, about starling flocks for a lot of biophysicists, is that you get this long-range correlation, you get this sort of stability even through this sort of perturbation. And you don't see a lot of damping, so you don't see a lot of decay of these flock formations, even when they're making these turns. Not much later, experiments on natural swarms of midges showed that the dynamical correlation functions display unmistakable traits of inertial relaxation. So this is where they're talking about these natural swarms of midges, which is a different system, and they have this inertial relaxation. So you can see the physical parameter in these natural systems. You can measure it, you can model it. At the same time, swarms have a dynamical critical exponent lower than any other reference ferromagnetic model. So these animal swarms, these swarms of birds and insects, they have these, you can actually compare them to materials in ferromagnets. And so this is a lot lower than the ferromagnetic model and even the Vichek model, which is actually for soft active materials. So this is interesting because you have these natural systems that you can model with this, you can approximate it with this model. You can approximate the behaviors quite well, but they behave much differently than, say, something like a material, something that's sometimes inorganic, sometimes organic, but they don't have this sort of maybe like individual cognitive components. In any case, all these traits prompt the development of a new model in which the inertial features of the dynamics, which in the Vichek model are washed out and averaged out, were reinstated, giving rise to conservation laws able to account for several new biological pieces of phenomenology. This model was the ISM, developed and further studied in 4 references here, the overdamped limit of which notably is the Vichek. The ISM model, this model that they're using, successfully reproduces several traits of natural flocks and swarms, including the linear propagation law of orientational waves in bird flocks. So this is where you have these bird flocks that move in a certain direction, they follow curves and turns, and they can maintain their stability. Even in density homogeneous systems, which are different types of bird flocks, the quantitative dependence, the quantitative dependence of the speed of propagation of these waves on polarization. 3, the precise inertial shape of the dynamical correlation functions in midge swarms. So you have this shape, and then the shape gets maintained as they kind of move around, maybe move around barriers or change direction. So you would expect some inertia there because of the physics, but you don't necessarily see that. So that means that they're making a lot of corrections sort of maybe before they even go into the turn or encounter these perturbations. They can really predict maybe what's happening at the front of the flock so it doesn't affect the back of the flock. You also have dynamic scaling laws, and then finally the precise prediction of the dynamical critical exponent in natural swarms. To our knowledge, the first case of agreement between theory, experiments, and numerics on a critical exponent of an actual biological system. So this is, they have a lot of equations here. This is the inertial spin model rule. So the key idea of the ISM compared to the Vichek model is that the social alignment force, which is the force that aligns individual birds and insects or whatever, instead of directly rotating the velocity of the agent, acts on its spin, which is the generator of the rotations of the velocity. In turn, the spin rotates the velocity according to these equations. So there's this relationship between velocity and spin, which is considered to be the same as between position and angular momentum in a ferromagnet. So they don't actually rotate, they're just like approximating the velocity and sort of the orientation of the birds. And then this corresponds to these different parameters, position and angular momentum, when rotations in the external space are considered. So the birds will actually change kind of their orientation as they're moving, like their pitch and yaw, or insects will do this as well sometimes. From this analogy, we clearly see that this parameter here, acts as a generalized inertia, a social or physiological resistance of the agents to rotate their direction of motion. So usually we assume in a lot of swarm or flocking cases that birds will operate on simple rules. So the Boyd's model of Reynolds, for example, it operates on these simple rules. You don't get too close to your neighbor, you don't get too far away. You follow their, directional change. And those are very simple social cues for the organisms. And this could work with cell migration as well. But it works. They're very simple rules. So there's this resistance. It's social, it could be physiological as well, where they rotate in their direction of motion. So this is where, as long as the interaction matrix is symmetric, the equation of motion for the velocity can also be derived from a time-dependent Lagrangian. So we can maintain this we can have this control over the inertial aspects. We have this, so this is sort of the inertial, which is set by these social and physiological resistance factors. And then, of course, we have, because of this, and if the interaction matrix is symmetric, we can use a Lagrangian, a time-dependent Lagrangian for this. The crucial point is if the interaction is symmetric under a given transformation, a global conservation law emerges. This is the case of the alignment interaction. The social force, F sub I, is invariant under the rotation of the velocity, so that once the symplectic structure of velocity and spin is restored, the generator of rotations, namely the total spin of the system, in absence of dissipative forces, constant in motion. So this is where you have this conservation law. In the ordered phase of the ISM, it is the cause of the emergence of linear spin, which is observed or spin waves observed in natural biological flux. So they go through some more of the math. They kind of model the stochastic components, this conservative fluctuation dissipation. As you can see, there is a lot of physics involved and these alignments with a biological system. And so we end up with this time-dependent Lagrangian, which is the discrete Laplacian. So we have this discrete Laplacian operator, which is here, and this is where we model system. And then we have this from noise on the sites to noise on the links. So it kind of talks about creating this network of a set of bird interactions, which result in a flock. So you have three birds here, I, J, and K. The connections A and B, so they form this network. And the network is between individual birds that are maybe observing each other or responding to each other in this inertial way. You have this adjacency matrix of this network. So we have for I and J, are I and J interacting? If so, yes, we have a 1. If so, no, we have a 0. So obviously I doesn't interact with I, and so forth. So we can model these networks of interaction as adjacency matrices. Then we have the discrete Laplacian, which kind of makes us into another, transforms us into a different set of values. And then this incidence matrix. So you can see that you have the adjacency matrix, the incidence matrix, and the discrete Laplacian, which form this very simple network. And this then, of course, can be plugged into this discrete Laplacian operator here for the discrete Laplacian. Okay, I think that's it for this paper. So, any questions about that paper? We have a thing in the chat here. So, okay, Dick asked another question. similarity to Ising models from atomic spins. And yeah, I think we talked about that, the Ising model. This is a specific case of the Ising model where they account for some of the inertial dynamics. Looks like we have Divanshu and Soham and Lalith. So we have some Aniketh. So we have like total turnover in our meeting. We had one group of people and another group of people. Okay, so I don't know if Aniketh or Divanshu or Soham or Lalith had anything they wanted to.
Sir, I have a small, vague idea of certain things I would like to discuss about.
Okay.
So I would like to share my screen and just give a short presentation of. Yeah. So I'm not pretty much sure about anything here in detail, but I just wanted to know like what I'm thinking of currently. Does it like really is a good fit for the this GSOC or not? So yeah. So first, I've been studying this. There is a work in the Nature Communications. I got the data that is related to embryogenesis of C. elegans. So it was for like 17 subjects, and for each subject there was start of this video. I mean like the movie from like the start of the like how the egg was started, and from there how it developed into a fully adult worm. And during this process, they analyzed using this principle where the cell membrane delineation, they have given this red with certain type of this thing, and the nucleus was like coded into green. So I think this is generally the dyeing process we add dye, and based on that this fluorescence mapping is provided. So they have given a proper data, the data for like all these files. And what I was thinking is, so based on this data, so first we can build a basic lineage tree. So when we do not have when we do not have where exactly the cell locations are, so the lineage tree has where it evolved. I mean like exactly the pattern, the way the C. elegans, the each and every nucleus have been evolved into a fully grown adult. We cannot know, but whereas with the data of the exact location, 3D locations, then we might know like what it has to do. And then we can, if we have like real-time nucleus data where exactly it is located, we can update this lineage tree accordingly. So I wanted to show, so suppose this is a lineage tree where the zygote is here, and from there it was, developed slowly, and it has been, each and every mutated, and later, so we have proper for like the 4 nuclei division. This looks like this. So this is an example where like 4 nuclei division. So we can map, you can find the distance between the each nuclei to nuclei, and the images that were previously used cannot give the accurate decision of the cell boundary, because cell boundaries are also important, because the way the membrane is structured, based on that we can determine certain characteristics such as the fate determination and the growth and the division patterns. So we can simply localize this nucleus based on certain standard deep learning methodologies, and based on that we can map back to the lineage trees, and then later on we can build by a good visualization tool determining the this application such as cell fate determination, growth division patterns, and probably spatiotemporal lineage tracking. So we can do that. I mean, like, I thought, like, this could be a good start off. This looks good to me. I've been seeing the data. The data is like rich and well. It is well structured. So I am thinking that we can apply different models into this space. Suppose we wanted to see how the lineage evolves, we can use hypergraphs exactly into the space where we can fit the coordinates and design accordingly. So I believe this data could be like useful in this exact space. So so this is what currently I am thinking, sir. That's it. I wanted to make it a more detailed, crisp presentation, but I probably need little more time for that, I think. So, yes, sir.
That's good. Thank you for going over that a little bit. Yeah, I think that's good. I did talk about the lineage tree earlier in the meeting, but
yes,
we basically have that. So I think with a lot of data sets that you would find, you'd have, with like microscopy data sets, you have to annotate the cells, or find out
yes,
they can be annotated. But once you have that, then you can follow those cells through their process, division. So yeah, that would work. And I think hypergraphs, of course, you can use that. I don't know if you read the preprint. I don't know if it's posted in the Slack channel, but there's a preprint that I developed originally, and then we were working on it last year. But this is where I kind of lay out a conceptual model of how to think about hypergraphs and lineage trees. So I'll send you a copy of that, or make sure it's re-upped in the Slack.
Yes, sir. I'll probably look into it in a detail, and I'll see like how this can be connected into that space.
Yeah, because that's like kind of a conceptual model. We can like use different data sets to kind of figure out
Justify
the idea.
Yeah.
Basically, the idea is you have, like, as we saw with the lineage tree, we have sublineages, or we have maybe like different types of tissues that form, and those would form the basis for the hypernodes. So hypernodes would basically be categories. So like if you had cells that would become neurons, we know this is C. elegans. We could put all those cells in the neuron category, which is a hypernode. And then those cells, sometimes cells move to another fate, sometimes they die. And sometimes you get, cell doubling, which means you get more cells, and their descendant cells are still in that category or not. So you have a certain number of cells in a category, then you have movement to other categories from developmental cells, say, which are a different category, and we can build a network like that where it's like in time, it evolves in time, but it also has this functional aspect where you have different cell types or tissue types that are sort of defined. So a cell belongs to this category. The category is a hypernode. The cells are nodes, and then they connect to other cells in other categories or hypernodes. So there's a hyper edge. Those hyper edges then are analogous to the hyper edges we find in a hypergraph. And we can do all sorts of things with that, because that's basically how a lineage tree is organized. It's organized by cell division, but then there's also this functional aspect. What are the cells going to become or doing right now, or what are they like? A lot of times in mammals, you have these multipotent cells where they can become, or what they call pluripotent, where they can become different types of cells. Sometimes they can become a range of muscle cells or a range of neurons. We don't know until they actually differentiate. But putting in these categories is useful because it can tell us from point to point what they're poised to become. So it's kind of an interesting way to do the representation. Anyways, I'll send you I'll make sure I re-up the paper. You can read through and
Yes, sir.
Yeah, see how that matches your ideas.
Yes. Okay, sir.
Yeah. Hi, Bradley. Am I audible?
Yeah.
Yeah. Hey. I'm sorry, I didn't know the meet time was shifted, so I joined a bit late. I wanted to share my idea regarding the project I was thinking on. So I've divided into three parts. The first one, it takes up the growing neural network work that we have done, and I was thinking if we could extend it to a hyper, like we have a graph network which is growing. If we could extend it to a hypergraph with the same analogies, maybe then we could capture some more relations between the cells. The second part was the work that is done till now has been specifically focused on C elegans. So if we could turn that work to a generalized library which could cater to any kind of data, that will be a more reproducible work. And third, I'm not sure if I will be having enough time, but if time permits, I was thinking to tap into the visualization part of the graphs, if that's something I could improve.
Yeah.
Yeah.
Yeah, I think we talked about this in the Slack.
Yeah.
So the first part, I think that's growing neural networks. That that was something that was forked from the neural developmental programs, and that's it's definitely needs more work in terms of making sure that that can recapitulate our lineage tree, right? Like, so we want to make sure that we have, like, every time a cell divides, we hit 2 new locations, that graph is like growing nodes and edges. And then, that's something that, of course, we can extend to a hypergraph where we have these functional categories, or we could have, I don't know, things within the cell that, there are different ways you can represent it. I would probably try, like, maybe like in the lineage tree, we talked about this, where you have sublineages. So like in C. elegans, you have like AB versus E1. E1, you have a bunch of sublineages, and EB, you have your sublineages. Just using those as categories, as sublineages, would help us with, like, in terms of visualizing the growing graph. So you'd have like a bunch of sublineages that form, their cells within those sublineages. And then so you have new hypernodes form when you get a sublineage forming, and then you have nodes within your hypernode complement increases with division methods. So you have these hypernodes that form, and then the hypernodes would stabilize, but then the hyper edges would increase. You can see it like that, which would be good in C. elegans because it's just a new way to visualize the lineage. And then we can do other things from that. I think expanding to beyond C. elegans is always useful. So we don't, the reason why we don't do that as often as we should is because it's extremely hard. Because all different, even if you go to C. elegans sister species, like C. Briggsy, that's another model. That's a very different animal. Literally, they're different. The lineage tree is different. There's a different nomenclature like that. So it's hard to kind of build a universal model. But we could do that, and I think it may be beyond what we could do in GSOC. Maybe we could build like a more general form of what we're doing, so we don't restrict it just to C. elegans. We have like a system where we have like a growing network, but something where you can assign the hypernode to some identity, and then the nodes are clearly cells, but then the hypernodes are maybe categories that you can just assign to anything, and then assign the nodes to some cells or cell division events, and then that would be like, because every organism seems to have those sorts of things. So that would be a way to tackle it as a and then finally visualization. So expanding beyond C. elegans, we want to be careful we don't get in over our heads. We want to make sure that we have the sort of general model. We'd have to work that out. And visualization, of course, is probably better. option in terms of a GSOC project for the summer. Like, we have the topological data analysis stuff, and that's, kind of vague in terms of how to view it. We have, like, you can view pretty pictures. Like, everyone puts, like, a UMAP in their paper, but it's hard to interpret it. So we have this opportunity to make these visualizations that can give you insight into what you're doing. So that's where, again, like visualizing the hypernodes, complement nodes, and then how they kind of, and then we have these layouts. And like, I don't know what the layout options are for hypergraphs, because those are a little bit different than the ones I was talking about earlier in the meeting for the Gephi plugins, because those are just for regular graphs. But that's something you might explore. Some of the Gephi plugins, some of the stuff for like Java's network tools or Python's network tools. You might explore that as well.
So should I try to do all three of them, or like prioritize on two maybe?
Oh, I'd probably prioritize the growing neural networks and then extending that to hypergraphs, and then the visualization stuff. And then the expanding beyond C. elegans might be something for later. So once you work out your methodology for 1 and 3, if 2 will be a little easier.
Okay. So I'll put that in the if time permits section.
Yes.
Okay.
Okay, yeah, so this is the preprint. This is on the archive. This is the one that this is the version, kind of the older version. There's a newer version. We haven't put it on the archive yet. This is with Mehul that I'm co-authoring it with. But basically, what I was talking about were these categories, and then this is the lineage tree here. So this is a hypergraph of an early lineage tree. It's not C. elegans, but it's very similar to C. elegans. It just works out the idea. So what we have here is we have the one cell, and then we get cell division, and we get this. Basically, we have the single that are, in this case, these gray ones are developmental somatic cells. And so there's a cell division event. You go from 1 to 2, 2 to 4, and then 7, 14, 28, because then you get this new category, which is the germ line, which starts at 1 and goes to 2, 4, 6, 12. So you get these hypernodes that grow in terms of their size, how many nodes are within it. And with each doubling event, we either can have a new category be born, some of the cells are assigned to that category, or we grow our original category by a certain number. So that's why these numbers increase. And then eventually we get to these differentiated cells like the connectome, which is these white categories. And again, we have some cell, this 28 here, this gray circle with 28 in it, you have a doubling event, which is doubling from 28 to 51. Well, it's not all doubling, but it's basically going from 28 to 51. 48 of those are still undifferentiated, and 3 differentiated neurons. So you get this doubling event, and then you assign all the cells from this 28 times roughly 2. You can have it. I think the idea is you're going to have asymmetric divisions, and then you assign those cells. So if you're looking at you're looking at a lineage tree, you get these events where the cells divide, and then you have to assign them to either the same continuity of the same hypernode category or a different hypernode category. And then, of course, cells can move from one category to another, or one hypernode to another over time. And then, of course, you have connections between them. So if you are looking at like their proximity or some signaling, you're going to have hyper edges which go between 2 hypernodes. And you're going to also have edges within that node. So that's kind of how this is supposed to be. And then there's another figure in here, which is where we add a spatial component. So we add in the spatial information of the lineage tree as well. And we look at that over time. So we have from a 128 cell to 256 cell embryo, these we have more of these hypernodes because there are more categories, and it's organized in terms of 2 spatial dimensions. But that's something, and again, we could do that with C. elegans because we have the spatial data. But I would keep it simple to start with and just try a lineage tree. I think that would probably working that out would be like a whole summer.
So I have a small question.
Okay.
So the given, the way the lineage tree develops, so we are trying to model it exactly, using hypergraphs and hypernodes. So when suppose like there is during the developmental process, if a parent, from the parent, it's split into like multiple childs, and these all do belong to a certain hyper edge. So, and from that we are, are we modeling it in a similar approach like the hypergraph does? It has multiple, we have hyper edge based on the given multiple nodes, and this edge is associated with a similar weight that is associated with certain number of nodes, and the lineage tree evolves based on the hypergraph structure. So while it's evolving, based on the lineage tree, so each and every node that is associated with individual cell and the spatial location of that cell. So we model the similar, suppose we have in first, it divides into 2 cells, and the 2 divides into 4 or 8 or certain pattern. So these all do belongs to a hyper edge. So based on like the way it's growing. So is it like that, sir?
Yeah. So a single cell will divide into 2 cells. And those 2 cells will have their own divisions, so 4 cells. And so the edges are, you'll have edges that are retained from the old case because the 2 new cells will maybe be in a similar location. All right, so let's say we have a cell here. It gives rise to 2 daughter cells, and then this gives rise to 2 other daughter cells. 4 cells. Now we have another cell over here. So in a graph relationship where we have 2 cells that are connected, those cells divide. There's daughter cells. So it's a growing network. We had 2 cells here, now we have 4 cells. These might retain their connectivity. This. And then if we think about it as a hypergraph, both of these might be in the same hypernode. So we have one hypernode that's unconnected to anything else. And then here we have maybe 2 hypernodes now, which have the daughter cells in them. So these 2 cells are in this hypernode. These would be A. These 2 nodes would be in this hypernode B. So we have A and B. And so there we have now a new hypernode with nodes within them, and they're connected through these hyper edges here.
Right.
And of course, these can be connected as well.
Yes.
And so we just grow in that manner. You don't always have to have a new hypernode. But,
Right.
Yeah. Do you see what I'm saying? Like each of these cells is assigned to a hypernode. So this is an A, A1, A2. This is A1, A2, B1, B2. So each node will change each daughter will change its assignment. It might retain its assignment or have a new assignment from the mother cell.
Yes. Yes, sir.
Yeah.
Thank you.
All right, well. All right, so any other questions before we go, or?
Yeah. Okay, so I had like 2 main directions for this year's GSOC ideas.
Okay.
So the one idea was regarding the growing graph neural network implementation that we have right now. The one thing that I noticed was it was more leaning towards the visualization, like essentially optimizing how the overall graph is structured with the data that we have. So it's not essentially like predicting anything new or doing anything new. So I was thinking of like, and coming to that, I was thinking of coming up with something similar to an ensemble of models to like given a starting number of parameters, maybe let the network grow and figure out how it actually evolves. So for that, there were actually I was actually going through the node features that I might be considering. Right now, the data set that is there in the repository, it has spatial temporal information, and so that's already there. And the other things what I was thinking was maybe like cell tracking, cell interaction, and the lineage tree as a whole. But like we did talk about this, I believe, last week, and you didn't mention that there aren't many data sets that are good for this, which has all the data. So we might have to end up stitching together data sets to get a complete idea. So, and for that, I actually went looking into the Riken data set, like from Japan. Let me just share. Yeah, so in that set of data sets as well, there were like a couple of issues. One being that most of the data sets is just one single entity or one single sample that's being studied. So you can't essentially like train a new model on it. So that's one thing I was thinking was I'm not completely sure of how feasible this is, because again, I'll have to be combing through a lot of data sets if we like go through the stitching together data idea. The other idea that I had was leaning more towards the actual visualization of the graphs and to try and create a UI or front end using Electron.js or something similar, so that you have a bit more control over how the graph evolves. So for example, you could like let the graph or let the model optimize how the graph is being displayed for a bit. Then you can pause the model and then make changes in the UI itself, give new, like maybe add new cells or give new positions to the cells and see how that evolves. So I believe that would actually be interesting for like the visualization aspect. So those were the two ideas I had. Yeah. Do you have any comments on it?
Yeah. So the first one about like having a visualization of the lineage tree and some of that. Last week I talked about the DevoWorm repository, and some of that was like some of that work has visualizations of the lineage tree in it. So I have some of the work that was done in previous GSOCs was pushed to DevoWorm, and some of it is in other repositories which I can point you to. I'll have to go look at it, but I remember in years past people have generated a lot of either benchmarks or work output, where they've like taken the sublineages of the lineage tree and they plotted them out in certain ways, and they like extrapolated the data, and those are available. I'll have to find them and show them to you, but that might be useful for the first part. We could build like a, the thing about like training a model on C. elegans, and you mentioned the work from the data sets of Riken, which are actually from published papers, but they have a repository. That most of you go across C. elegans, the lineage tree is mostly stable. I mean, there isn't a lot of variation. It's not really of interest biologically. So really, if I take a single C. elegans and those data that you mentioned also aren't just a single worm, but like it's basically you get, lineage tree. So the lineage tree is, has these certain properties. It's actually a pretty simple lineage tree, comparatively speaking, compared to other types of organisms, but it does have the structure we can understand, and we do have the data that can build like a model of the lineage tree. So one of the things we can do is we can vary that lineage tree in different ways. We can build a continuous model of a lineage tree, and I think we have some modeling exercises in some of the previous year's GSOC repositories, which are not in DevoWorm, but I'll have to bring them up and kind of maybe I'll bring them up next week so you can see them, or I can send them to you. You can look them over. But I think we can do that, and that's sort of these things where they normalize their data, they produce these cool graphs, but then it doesn't really do anything beyond that. So that would be a cool, kind of a nice thing to kind of take that and run with it and see what you can do. Maybe we can, because we've talked before about simulating lineage trees, and we haven't done that in a way that was like something we could publish or something we could like say anything about. So, I mean, that would be an interesting thing. And then I think the visualization of graphs is always good. Like I was saying, I was talking, I think, to Aniketh or Soham about it. This is something we can work out with different tools. I think Electron.js might be useful for having an interactive form of it. Like, say, I want to plot it in this way or that way. I want to visualize it. But, you can do a lot of that in Gephi, too. And that's all the, Gephi is kind of like the GUI, and then you can actually, build extensions into that. So, you'd focus on kind of the layouts. But yeah, we'd have to talk a little bit more about that in terms of what might be reinventing the wheel and what might be really useful.
Yeah, okay.
All right. Well, thanks to everyone for meeting today, and hope everyone has a good week. Yeah, see you next week.
Okay, bye.
Okay, bye. Bye-bye.